Hello everyone, bonjour tout le monde. Mon nom c'est Shelley McCoy. My name is Shelley McCoy, and on behalf of the Foundation for Conservation of Atlantic Salmon, I'd like to say welcome to today's webinar. Alors, bienvenue aujourd'hui au début de notre série des webinaires. For today's webinar, we're really pleased to be hosting Alexandre Carbonneau. He's currently a PhD student at Laval University. Until last fall, Alexandre was supervised by Louis Benachet until his passing and co-supervised by Julien April from the Minister of la Faune. He's now supervised by Jean-Pierre Tremblay and still by Julien April. He comes from a small town in the eastern sections of Quebec. Alexandre took a technical course at the Cégep of Sherbrooke, where he focused on the practical side of biology, and then went on to do a bachelor's degree in ecology at the University of Sherbrooke. Then started a master's degree at the Université Laval with Louis Benachet, who then suggested a fast track to a PhD, which he quickly accepted. Today, his main interest is in species management and conservation, and he uses genomic tools to inform managers and help them make the most informed decisions possible. So today, Alexandre will be speaking on genetic population structure of Atlantic salmon in Nunavik. So the presentation today we decided to do in English. However, we do have AI translation. So in the chat, you'll see the instructions there on how to access that. So you can listen to the presentation today in English or French, it'll be your choice. So to no further ado, I will pass the presentation over to Alexandre. So Alexandre, thank you and welcome to today's webinar, and the floor is yours.
Thank you. Good afternoon, everybody, and thank you for the opportunity to present this afternoon. Oh, I touched something already. Sorry. What did I do? I don't see my screen anymore.
You see it, Alexandre. I see it on here.
Okay, perfect. There was something on my screen. So again, good afternoon, everybody. Thank you for the opportunity for presenting my first chapter of my PhD. As Charlene said, I did this work supervised by Louis Benachet and Julien April, and it was part of a huge project called Fishes that I've been working on. Just give me a second here. Okay, so Fishes project for fostering indigenous small-scale fisheries for health, economy, and food security. And what I'm presenting in this slide is true for pretty much all across northern Canada, but I will be focusing on Nunavik since I'm working on Atlantic salmon, and Atlantic salmon only occurs in Nunavik when we talk about northern Canada. So as many of you might already know, in Nunavik, high prices in grocery stores and poverty level lead to a lot of food insecurity. And because of that, and also because of cultural importance, Inuit rely a lot on wildlife to eat. The issue here is that the lack of knowledge on these harvested species can lead or potentially lead to a decrease in harvested species in terms of population size, which increase the food insecurity issue. So you see the loop here. So the overall goal of Fishes project, and I would even say in a more broader sense in conservation in general, is to improve our knowledge and thus the species management. And when we're talking about northern community, in order to maximize food security and cultural knowledge transmission. So I kind of already introduced that, but the overall goal of the Fishes project was to combine scientific and indigenous knowledge on 7 species that are fished in Canada's northern indigenous communities, especially only actually in the Inuit, Cree, and Dene communities. So I will be today focusing on Atlantic salmon and only talking about Nunavik in northern Quebec. So to introduce...
Just to put te déranger, là, we only see part of your screen. So I just want a heads up. Just savoir.
Maybe it's because of that. I think I touched something at the beginning. Is it better?
Yeah, on voit comme 75%, 75% of your screen right now. So.
Ooh, that's weird.
Yeah.
Just a second. Stop share. Or restart sharing.
Already looks better. Perfect. Okay.
Oh, okay. I'll try not to touch too many things just to make sure it stays like that. So yeah, to introduce more about Atlantic salmon in Nunavik and mostly in Ungava Bay, I will make appear in a few seconds the different rivers, but I just wanted to say that we already knew before the project began that there are 7 salmon rivers that flows in Ungava Bay and 1 river in Hudson Bay. And we already knew that there was at least 3 genetically distinct populations, which are the Feuille here river, the Coxoac here circled in red, and the George River. The issue here was that we were lacking a lot of information, mostly genetically speaking, on the watershed inside of the Coxoac River. And same thing here for the Baleine and Nestapuka River that flows in Hudson Bay. I just wanted to make a quick saying about the Coxoac River. I will be talking about it a lot along the presentation. So just to let you know that I'm talking about the river in the middle that is circled in red. Another important information about the Nunavik Atlantic salmon, especially in the Coxoac River, is that there are 3 different life history migratory tactic, and probably, as many of you know, landlocked salmon are salmon that spend their entire life cycle in fresh water. Then there are also the marine ecotype, the marine tactic, which are pretty much the typical anadromous salmon that we see in the south of the country, which means they migrate into the ocean to feed and then they come back to spawn in their river. What's exceptional here, especially in the Coxoac River, is the presence of an estuarine tactic. So these individuals never reach the ocean. They migrate into the estuary, they feed there, and then they come back to spawn in their river. But we had, again, no genetic information about these tactics. One maybe last information in this context part of the presentation is the presence of likely the Nunavik's most important subsistence salmon fishery that occurs in the Coxoac River, which is represented here by the blue square. And again, we had no information on the origin of the fish that are caught in these very important subsistence fisheries. So we had no information where the fish were coming from. So in this context, our goal was first to establish the extent of genetic differentiation between Nunavik's salmon rivers, and also between estuarine and marine migration tactics that are present in the Coxoac River. And the second goal was to determine the contribution of populations and migration tactics in the estuarine Coxoac subsistence mixed stock fisheries. To do so, we collected adipose fin in order to get genetic data in all of the 8 studied rivers. And we also collected scales for tactic identification only in the Coxoac River. We had at our disposal between 19 and 42 individuals in all of the Ungava Bay rivers, and we had 10 individuals in the Nestapuka River, which is on the Hudson Bay side. We also had 280 individuals that were coming from the subsistence fisheries, from the mixed stock fishery. And all of these 280 individuals were samples, were collected from, by Inuit community members. It was important for us because we wanted this sample to be as representative as possible to the fishery, the subsistence fishery that occur there. We then extracted DNA. We sequenced DNA using genotype by sequencing, and after filtering steps, we had just over 14,000 SNPs, and results are based on these data. So I will be first focusing on the first objective, the first sub-objective. Let's call it this way. And we identified 7 genetically distinct populations out of the 8 rivers. So in the next slides, I will be explaining and detailing every or almost every rivers and all populations. Every time I'm presenting this PCA, this principal component analysis, in the middle it's a mixture analysis, and we have the FST values on the bottom of the figure. I'm also always referring on the map, just so you can visualize more what river I'm talking about. So let's start with the Kenya-Pisco River, which is here, as we can see on all analysis, clearly genetically differentiated from every other rivers. I forgot to mention that each dot, for those of you maybe who are less used to it, each dot on the PCA are individuals, and each bar on the admixture plot are also individuals. So yeah, you can also see that on the PCA, there's a lot more variability in the Kenya-Pisco River compared to other rivers, other clusters. And this is likely due to unidirectional gene flow from an isolated population upstream from an impassable fall. So what happens here is that we know that there's a population here, a landlocked population here above this fall that is represented by the triangle here. And all of our samples were coming from downstream from this fall. So this tiny part that is about, I think it's about 20 or 23 kilometers in this river. So we know that the population here, at least a few individuals from this population migrate downstream from this fall, and then they can't come back upstream from this fall. So this brings gene flow, it brings genetic variability, and this is what we can see here on the PCA. Then talking about the Nestapoka River, which flows in Hudson Bay. Again, clearly differentiated, as we can see on all analysis that I'm presenting here. What's very interesting here is that the differentiation with Dugay, Delay, and Mélèze River. So I'm talking about the 3 rivers here. Mélèze is in light green, Dugay in pink, and Delay in purple. The differentiation with these 3 rivers are weaker or lower. And this is in accordance with an hypothesis that was proposed in early 90s that the Nestapoka River was colonized by one of these 3 rivers, or maybe by these 3 rivers, we don't know. And this makes sense with our data, since the differentiation with those rivers are weaker. Then I will be zooming on the 2 clusters here that we can see on the PCA, just so we can see a bit more the results, which give this new PCA. And I won't say much on these 2 rivers, except that they are, again, clearly differentiated. We know that the gene flow between the sorry, looking for my cursor. We know that the gene flow between Coxsackie River and Baleine River is quite high, which makes sense because they are geographically close from each other. But then talking about gene flow and geographic distance, we have here Feuille and George rivers, which are genetically differentiated, but less than what we expected, considering their quite high geographic distance. And this similarity we think might be due to similarities between watersheds, mostly in terms of habitat and water temperature. So this might increase the chances of straying between those two rivers, and thus gene flow between the rivers, and ultimately the genetic similarity. And to finish on this first objective about the Duguay and Delay rivers, which are the two rivers that we couldn't clearly genetically differentiate. In all analysis, they appeared pretty much the same, genetically speaking. And I mean, in all analysis I'm presenting here, because I have an analysis that I will be showing in just a few slides that shows an assignment success in these two rivers of 87%, which suggests that they are not, although they are very similar, they are not completely panmictic. And we suggest that demographic analysis should be done in the future. Just a cool, quick result here showing that there is an isolation by distance effect. So a significant effect of geographic distance on genetic differentiation with an adjusted R-square of 0.39. So it basically means that the further apart sites are, the more genetically distinct they are. I just want to point here the Feuille and George comparison with a quite low genetic differentiation for the great geographic distance here. So it just suggests that something else than distance only is explaining this genetic differentiation. Then I'll be jumping on the second sub-objective of the first objective, which was to differentiate the life history tactics. For this, I used all of the 72 individuals that we caught within the Duguay and Delay rivers, and I added 200 individuals that we assigned, that we captured in the Coxsackie estuary, and then we assigned to these two rivers. I'm using these two rivers because of their nearly panmictic structure, and I didn't want to study the difference between rivers, so I didn't use Mélèze or Kenya-Pisco individuals because it could have kind of blurred the signal in the analysis. Then here, as we can see on this PCA with all of these individuals, and same thing here with the FST value, we found no genetic differentiation between those tactics, at least at the population scale. However, I've been able to identify SNPs that are associated with migration tactics, especially on chromosome 2 and on the chromosomal fusion between chromosome 8 and 29. And some genes that were located close to these outlier SNPs were linked to various biological functions that may have an impact on the timing of smoltification. So the biological function were growth, response to temperature, brain development, and light detection. And although we didn't find necessarily more biological function than that, Kyle Wellband identified more biological function in this same area in the genome. What's important to understand here is that, well, if a salmon migrate into the ocean from the Coxsackie River and it goes to Greenland, which is around here, or in the Labrador Sea, it obviously have to go through the Ungava Bay. And there was an hypothesis in early 80s from Power that suggests that there's an effect of water temperature in Ungava Bay. So it means that early migrant within a season, and I'm talking here about smolt, so I'm talking about salmon that are going to the ocean for their first time in their life. Early migrant in the season are able to reach the ocean because water temperature in Ungava Bay is relatively high. And I say relatively high because I mean it's still north. The water temperature is still low, but well high enough for the salmon to go through the Ungava Bay. What happens? Well, based on this hypothesis, what happens with estuarine salmon is that we think that there might be later migrants. Again, talking about smolts here. So they can't reach the ocean because the water temperature is too low in Ungava Bay. So they kind of stay stuck in the estuary. And this hypothesis that Power advanced is in accordance with what we found, since we found a few genes close to our SNPs outlier that were linked to biological process that were linked to smoltification. And this is in accordance with the principle of indirect genetic control, which very quickly explained means that the genotype of individuals have an impact on physiological condition. Let's talk here about things like growth and well, the effect of temperature on these individuals, which then have an impact on timing of smoltification and then have an impact on the adapted migratory behavior. I'm not talking much about the environment here because we couldn't test it, but it's likely that there's an impact of it. And this also have been documented in many other salmonids. I will be jumping now on the second goal of the project, which, just as a reminder, was to determine the contribution of populations and migration tactics in the estuarine coxswain subsistence mixed stock fisheries. To do so, I mean, to be able to assign individuals that were caught in the estuary to their river of origin, I had to demonstrate the power of our data set to do so. So I used all of the 246 individuals from source populations that were caught within each rivers, and I ran the analysis, and I got an assignment success of at least 93%. The only exception here was the Dugay and Dele rivers with 87–88% here in the Dugay. But this is the analysis I was talking about earlier that shows that we are able to assign individuals in these two rivers with a quite high assignment success, and it suggests that they are not completely panmictic. Just so you know, when I ran the analysis by combining Dugay and Dele rivers, we had an assignment success of at least, not at least, but an assignment success of 100%. So this means that if there are bias, if there are an individual that is wrongly assigned, it should be between those two rivers, because there was a 100% assignment in those two rivers. I then assigned individuals that were caught in the Coxswain estuary, and I discovered that 100% of individuals that were caught in the estuary came from within the Coxswain River, and over 80% were coming from those two rivers, Dugay and Dele rivers. So why those two rivers and not Mélèze or Kenya-Pisco? Well, we think that it's mostly because of higher habitat quality in those two rivers compared to Mélèze here in light green and Kenya-Pisco. And as I said earlier, the habitat available for anadromous salmon is quite small in the Kenya-Pisco River. So there's a small habitat and also a weaker habitat quality in the Kenya-Pisco. We also found no interannual variation in the origin of these fish, meaning that there was pretty much the same contribution of each river each year, at least for the 2 years that we had. I then did the same thing for migratory tactics, and we found a major contribution of both estuarine and marine tactics, meaning that they both contribute in a pretty important way. There's not one tactic that contributes super high and the other one not. And we also found a significant interannual variation in their contribution, meaning that we had significantly more estuarine salmon in 2020 and significantly more marine salmon in 2021. So in conclusion, it went faster than I thought. In conclusion, we found 7 genetically distinct population out of the 8 studied rivers. And as I said, Duguay and Delay rivers are possibly demographically independent, but obviously more studies will have to be done. We still suggest to remain cautious and to manage those 2 rivers as 2 distinct populations. And this suggests that the what we call the river by river management plan that is already applied by the Quebec government is appropriate. About the migratory tactics, we found no genetic differentiation between estuarine and marine salmon, at least at the population scale, but we found an intra-population genetic polymorphism. This implies that life history tactic must be managed as intra-population diversity, and it's particularly important since these tactics might have helped the species to survive in this harsh environment. And finally, as I said, 100% of individuals that were caught within the Coxswack estuary were coming from within the Coxswack River, and over 90% were coming from 2 rivers, so Duguay and Delay. And the contribution of both tactics was major and very annual. So this implies that Duguay and Delay rivers must be management priorities, especially since they represent the production of the vast majority of salmon that are caught in the Inuit Kudrak fisheries in the estuary. And also because a majority of outfitters in the system are located in those 2 rivers. And it is all the more relevant given that no fish were caught from outside of the Coxswack watershed, so from Feuille, Baleine, or George rivers. So I want to thank you all for listening to me. I want to also thank my 2 supervisors, Louis Bernatchez and Julien April, as well as all of my co-author, Makivik and Kudjamiut, who helped us a lot with the sampling, and Genome Quebec and Canada for the financial part. Just wanted to share with you the paper that was published last winter, and also my email address if you want to send me any question or anything. We can chat about it in the next days or week. It will be a pleasure. Thank you.
Merci, Alexandre. Thank you so much for the presentation. And now we're ready for questions and answers. I would encourage you to raise your hand so I can unmute you so you can ask your own question. If you have any difficulties with your system to do that, you can always type it in and I can read it out loud, but you take the chance of me reading it wrongly. So I encourage you to raise your hand, but either or will work. So the floor is now open if there's any questions. We've got a quiet group today. In a minute.
I haven't been able to see if I know anybody, but I've been presenting these results a few times, so maybe maybe a few of you already heard.
Well, we get a variety in the audience here. Well, if someone thinks of a question afterwards, which happens a lot to me, once you have time to digest the information, as Alexandre has his email, and I asked him beforehand if he minded anybody, sending directly a question to him, and he said no problem. So you can do that if you do have a follow-up question at a later date. Please feel free, or you can send it our way, and we'll make sure he gets it, and we'll try to answer. In the meantime, this is recorded, so it will be available on our YouTube page. So for those folks that were not able to join us today and would like to listen in, they can always access it that way. And I'll also mention that Alexandre is the first presenter out of 10 throughout this series. We have another presentation coming up on November 6th. It's with Sarah Kingsbury from DFO, and she's going to be speaking on a new tool for setting biodiversity management priorities adapted from aquatic invasive species management. It's a pilot using Atlantic salmon in Nova Scotia. So please feel free to register for that and join us for that event. But for now, I just want to say another special thank you to Alexandre and for everybody joining us today. And I wish you all well. Thank you.
Thank you, and thanks a lot for the opportunity.
You're welcome. All right, see you, everybody.